So Chris is out this morning, so he asked me to introduce myself, so allow myself to introduce myself. I'm Ben Neale. I'm at the Analytic and Translational Genetics Unit over at MGH, and I'm an institute member here at the Broad. And I'm going to talk to you about the analysis of rare variants from sequencing studies. And really, this talk will be focused more on how we think about study design, how we think about testing, how we think about the kinds of ways that we'll actually evaluate the rare variants that might come out of a pipeline that, say, Geraldine talked to you about in this series a little bit earlier. And I'm really going to do a deep dive on this paper in particular. So this is a paper that we wrote with Eric and Orr a few years back, really in a sense to set the scene for how we think about rare variant association studies, what are the kind of various parameters in play, and how do we think about really the identification of genes from large-scale exome and genome sequencing projects, in particular for complex traits. So this is much more focused on phenotypes like diabetes and schizophrenia, blood pressure and height, rather than being focused on some of the more rarer and Mendelian-like phenotypes in the scheme of things. Okay, so as with many genetic and genomic studies, we have a variety of goals, but one of the kind of primary goals and real major driving and motivating factors is this idea to try and understand the genomic basis of disease. And understanding the genomic basis of disease gives you a lot of potential avenues of interest and exploration. It motivates novel biological hypotheses. It can teach you things about epidemiology. It can teach you things about causal risk factors and how different parts of the biological system relate to one another. All of these things are kind of on the table as end product results from genetic and genomic studies. But the one that I think is of particular importance, and they're all important, but the one that is of particular importance is this idea of gene identification and really using that to drive forward novel therapeutic hypotheses. I think there's a lot of excitement and enthusiasm about the prospect of that arc, and that's been very much the case for cancer in terms of genomic technologies really driving forward therapeutic interventions there. And I think the hope is that this can really start to play out for complex traits as well. So Eric being Eric, rather than calling GWAS a GWAS, he's like, well, it's not actually a genome-wide association study insofar as it's actually a test of all of the common variants, and so we should really perhaps call it a common variant association study rather than a genome-wide association study. And then there's this contrast with this rare variant association study. And there's a kind of natural question that, kind of arises when you think about this distinction between evaluating and interrogating common variation for, risk for disease versus rare variation, and it's kind of what's common and what's rare. And I think people are fond of saying something like 1/10%, something like that, or 0.5%, or maybe 1/100th of a percent, depending on your sample size. And I think that's really the kind of key, is that the sample size is the major indicator as to whether or not we'll have sufficient information from a statistical point of view to really test a individual variant. So if you're sort of, below, say, 40 or maybe 80 copies of the minor allele, that's probably a reasonable rule of thumb for declaring something rare. So if I have a sample size of 400, rare might not be very exciting at all. It might be just, in a sense, 80 out of 800 chromosomes is, like 10%. So that's not really rare, but that's just kind of what we can test properly. If I have a sample size of, 400,000, then that kind of dramatically changes what the nature of rare is, and I think that's an important thing to keep in mind. Now, it's also worth flagging that, the primary engine of CVAS, kind of GWAS chips, genome-wide association chips, they work reasonably damn well with, if you kind of throw in imputation, they work reasonably well down to maybe 1% or 0.5%, depending on your reference panel size, like how well can you impute and how dense are your markers and those sorts of things. So there is a sort of technological limit that's also imposed on this, because unless you transition to a sequencing study, you're not really going to be able to look at the things that are, fundamentally much rarer than that. Okay, so let's think about CVAS versus RVAS. Well, with CVAS, it's all very straightforward. You simply pick a variant, pick a SNP, copy number variant. It doesn't matter, whatever. Choose your variant that's common enough in your sample to test, and then you just simply compare the rate of that in cases versus the rate of that in controls. It's a very straightforward study design, really, really intuitive. Actually, when GWAS was coming on the scene, there were people that were maybe suggesting that the statisticians would get bored running basically a t-test. I can assure you the statisticians did not get bored with lots more data, but there was the concern that this was in a sense too simplistic. Now, what's also nice is that you get a direct estimate of the effect size, right? So you perform this association test, and you calculate the effect size explicitly. It's a very straightforward calculation. You're only looking at a single variant. Now, with rare variant association studies, we have quite a lot of decisions to make. Okay, so we're now no longer in this regime where we can do a direct test of every individual variant. The number of copies is far too few, so we're short on information. Our association testing paradigm from common variant association space isn't going to work very well. So we're going to have to adopt a somewhat different strategy here. We got to think about aggregating multiple different mutations. We can kind of think about looking at, LOFs or missense mutations, or maybe some combination of the both. We may want to think about how to filter those mutations. So maybe if we knew all of the ones that were actually likely to cause disease or likely to influence disease, and we had a really good way of divining that, we should maybe include that information in the way we perform and calculate our test. And that's a kind of structural difference from what we're doing in common variant space, where we don't care, we just go along and test everything as we go. And then we're going to do the same comparing the rate in cases versus the rate in controls. That's going to be the exact same kind of approach in general. But rather than having a direct estimate of the effect size, right, because we're grouping variants together, we're not going to make some inference about what the effect size might be. And really it's a guess, and it's a kind of bulk-wise guess. It's not like I know exactly that this variant is going to have an odds ratio of 2, or this variant is going to have an odds ratio of 3. And that's a real structural change in another way, because it means that, figuring out what the phenotypic consequences of the rare variants that you lump into your test is going to be more complicated. But it does also hold open the potential that you might, get at something that we maybe call in other domains like an allelic series. So something where we have, a variety, a spectrum of mutations that have differing effect sizes, and that range of effect sizes can then be used to, potentially, if you have a large enough sample size, kind of validate that range of risk that you might observe. My current favorite example of that would actually just be the C4 copy number that Steve McCarroll and Ashwin Sekar really did for schizophrenia, and showing that kind of pretty much dose-response kind of relationship between the amount of C4 gene, because it's copy number variable, and the risk to schizophrenia. So that's a really nice model, and it's really hard to get that if there isn't a kind of good relationship. If you have this dose response on risk, that definitely gives you more confidence in the validity and the veracity of the finding. Okay, so there are a bunch of questions, like what variants do we want to include? Do we want to include loss of functions? Do we want to include missense? What about synonymous? We probably don't want to include those, but it's on the table. Like someone, a lot of my geneticist colleagues say, never work on synonymous variation. It will lead to career ruin, because there have been a lot of times where people have tried to claim very, exciting and novel things about synonymous variation, and then it doesn't really pan out because there's some error in the data or bias or whatever. We can also think about what frequency threshold, and really the goal here when we think about the frequency threshold is what is going to drive power for association. Remember, our goal here is to identify genes that drive risk for disease. And it is always worthwhile to remind yourself why you are engaging in these kinds of studies. And front and center, the goal for this kind of approach is really the identification of specific genes for novel discovery. Now, there are lots of other things that you can do with lots of different kinds of genomic data. But if you're going to dress up your work as making an impact on disease, then you have to remember that maximizing power to find association is a really important and critical component of actual study design. It's all well and good to have, idealized notions about how exciting genomes might be, but if you can do 3 times as many people with exomes, that might be a preferable study design given the state of play. Question of sample size. Well, again, when we think about sample size, that's related to this idea of power. So what we're going to try and do in this is kind of think about ways that we can structure our study designs and use that, kind of conceptual framework to scope out what might be required to, actually find the genes that we think are underlying risk for disease in the population. And so sample size and power are kind of, nicely related, and there's a fair amount of nuance there. There are what I might call upsides, things that we can do that might improve our power to detect. Again, we're driving at this idea of finding genes being our core goal, and so if we can maybe be opportunistic and focus on population isolates or maybe on de novo mutation. I'm going to talk about the isolates. I won't talk about de novos. I think Caitlin will talk about de novos in a little bit. In this series, but these kinds of different ways, different demographies of populations may change and impact our ability to actually identify genes that confer risk to disease. And then there's this question of genomes versus the non-coding, and the way that we're going to address this is we're just going to try and build a model. Okay, so this model is not a perfect representation of the way the world is, but it should be reasonably instructive for the kinds of decisions that we should be making when we're engaging in these kinds of studies. Okay, and it's always worth reminding yourself that it's a model as well, that models have lots of advantages. We can do them computationally, which is very cheap, right? So a few electrons isn't so expensive. And so you can kind of build these potential, in a sense, power scenarios and try and represent as much as we possibly can about the way genetic variation operates in the population to try and make some inference about how we should design our studies, what we should be trying to drive at when we think about these kinds of approaches. Okay. So the genetic model that is, in a sense, most important is what is happening with respect to risk. Okay, so how are the genetic variants that are influencing risk for disease, how are they, operating in the population? What do they look like? How do we think about, modeling the kind of risk landscape? And the approach here is to just take what we would describe as a very simple model. So we're going to have this model there. There are basically 2 classes of mutations. There are things that confer risk, and then there are things that are neutral. Obviously, this is a simplification. We expect a spectrum of effect size. It is potentially possible that variation within a gene moves in opposite directions, and so on and so forth. There are lots of ways that this model can break down, in a sense. But this model itself should capture some chunk of reality, we hope. Okay? Now, our primary engine of association that we're going to deal with here is really burden tests. And all that burden test is doing is saying, what is the number of alleles that are in cases, and how do we contrast that with the number of alleles that are in controls of a given class of rare variation? Okay, so it's just counting and comparing the frequency. And, a lot of our job is counting. That's okay. It's kind of important. But the things that really matter when we think about these sorts of alleles and these kinds of mutations and these sorts of phenomena is how much variation are we expecting to see in the population? How much variation is there going to be in a sample if we go out and collect, a couple thousand people worth of cases and controls? How's it going to shake out? Well, the major driver of the amount of genetic variation in the population is the mutation rate. Right? So we have this, wonderful process of mutation, newly arising mutations occurring every generation. You have new mutations that your parents don't have. I absolutely guarantee that. And we can think about a kind of typical gene. Okay, so this is all on John Q. Public gene. So an average size gene of around 1,500 bases. And then we can look at what the mutation rate is for different classes of variation. Okay, and that's going to govern how common certain kinds of variation really are. So we can think about our silent or our synonymous mutations, and they have, some mutation rate overall in the population. And then the disruptive ones are LOFs and our essential splice sites, and those kinds of mutations. They're less common in terms of footprint, so there's a lower probability of those kinds of mutations occurring generationally. And then you can think about missense mutations, and this is the lion's share of the kind of genetic variation mutational landscape, right? So there's more opportunities for a missense mutation in your typical coding region for a gene than there are for, say, silent mutations or disruptive mutations, okay? Now, the reason that I flag this is not because we're going to engage in a huge amount of de novo sequencing and trio sequencing and looking for de novos. It's because the rate at which these occur is directly related to the rate at which they accumulate in the population. Okay. Then the second piece. So on one hand, we have our engine of mutation arisings. This is kind of more population genetics in a sense. So we've got these newly arising mutations that are coming in, and then on the other side, we have the purifying selection force, okay? So the kind of natural selection force that says, hey, you, Mr. Mutation, you seem to have a negative impact on fertility and fecundity, and as a result, you stand less of a chance of persisting and propagating in the population. And so what purifying selection is doing is it's taking those negative alleles and it's pushing them out of the population because there's a reproductive disadvantage for the individuals carrying those kinds of mutations, okay? And this is the core concept behind a huge amount of population genetics, this idea of mutation selection balance. So our two-class model is that the neutral variation has no selective coefficient. There's no, impact. It's a neutral allele. It's just going to wander around as neutral variation is wont to do in the population and be a consequence of forces like genetic drift and population demography and those kinds of things. And it does nothing with respect to risk, so there's no excess risk conferred as a function of this. And then on the other side, we have these null mutations. And I'm using null here to kind of telegraph the LOF, the disruptive kinds of ideas. But this model you can, kind of pervert a little bit if you want to think about only genes where there are missense mutations that would increase, the kind of gain of function kinds of mutations. You can kind of, tweak it to get there. But we still have reasonable expectation that there should be a fair number of disruptive mutations in genes that are conferring risk across a variety of different complex traits. And then we have this idea of excess disease risk, this lambda, this boost on the kind of rate of disease in an individual that happens to carry one of these mutations that we put into this null category. Now, our silent mutations, we're saying, are all neutral. That's very, it's just, again, it's a model, it's an assumption. There are almost certainly circumstances where that's slightly broken, but it's not so bad. And then our disruptive mutations, these putative loss of function mutations, these premature stop codons, frame shifting insertion deletion mutations, those kinds of variants, we are going to say, are all null. And that's one of the nicest things about working with LOFs in general, is that the expectation is reasonably strong that the effect will be lined up for all of those kinds of mutations. It will be exactly the same kind of effect. And then these missense mutations, this is where we're going to now impose like a little bit more nuance. So if you look at the evolutionary conservation literature, okay, so you do comparison of species and you look at the number of fixed differences and things like missense mutations, there is clear evidence that some missense mutations are definitely very bad, and some missense mutations are basically neutral. Okay, so there's this, in a sense, this range of possible outcomes for missense mutations. For the sake of simplicity, we are imposing again this two kind of class model, and we're going to say that some fraction, say alpha, and you can kind of squabble about what alpha is, but some fraction alpha will be equivalent to a null mutation. And then the remaining fraction, the 1 minus alpha fraction, is going to behave like the neutral variation. Okay, so that's just the setup itself. Now, for the typical gene and what we see when we do the kind of comparative genomics and conservation and those kinds of studies as a community, we see that maybe the alpha is around a quarter. And that's, again, a model. It's part of the model. There are, of course, genes where alpha will be much higher, much closer to maybe 50% or even 75%. Genes where missense mutations are really, really bad, pretty much in general. And then there'll be genes on the other side where missense mutations don't really matter at all. that it's really only loss of function mutations, and the fraction of the variation is quite small. But this is not an unreasonable average based on what the comparative genomics is.
So does this pairwise selection also apply to any mutations that would be beneficial for the population?
So for beneficial mutations, it comes with a very different set of considerations. So rather than having kind of natural selection working the mutations out of the population, what happens is those mutations wander around at very low frequencies for a very long time because they don't really gain traction. They don't get common enough to really have an impact on selection. But then once they get to be common enough, like 1 to 5% or so, then they really zoom up the allele frequency spectrum because they're now common enough in the population that they're actually having an impact at a sufficient level. And then what ends up happening is that they zoom up in allele frequency, and then selection gets more and more inefficient as it gets rarer and rarer in a sense. And so what happens is the alleles flip, and so it now becomes a rare allele, but on the derived side rather than on the inherited side. The more complicated scenario is what we might describe as balancing selection, and that does all kinds of weird and complicated things, and you can read about all of those crazy dynamics if you want to go into the deep dive in the population genetics literature. Okay, so back to our model. We've got our neutral mutations and our neutral mutations and our null mutations, and now we have this addition of missense mutations that we're going to kind of layer on top. Okay, so key figures of merit that we can think about. The first and foremost is combined allele frequency. Okay, so this is basically the sum total of the frequency of the mutations that fit into a given class in a given gene. Okay, and that's very appealing as a figure because remember what we're going to be doing is testing the rates of mutation in cases versus controls, and so the combined rate of a class of mutations in a gene is exactly what we would want to test ideally. Okay, now there are a couple of nice kind of population genetics rules of thumb. Okay, and the rules of thumb are that things that are under weak selection, the combined allele frequency is a function of the effective sample size and the mutation rate. Okay, and this NE, this N, that N there is the effective population size. It's not population size right now. This is our, approximately 10,000 population size sort of figure. And for weak selection, the population size, 4 times the samples, times the effective population size times the mutation rate will be what we would expect to see for a combined allele frequency. Okay, so there's this general rule of thumb. So this is what we think of as our neutral model. Then we have strong selection. So these are mutations where S is quite large. So that is to say that the mutations are being worked out of the population very rapidly and very aggressively. And in that kind of circumstance, we transition to this regime that is basically mu over S, okay? Now, the most intuitive way to think about mu over S is to think about what happens if a mutation knocks out fertility altogether. So if you have this mutation, you are sterile. That means that mutation is only going to exist in you. You have no opportunity for having progeny. There's no way to, give that mutation on. And so under that circumstance, S is now 1. It is complete knockout, which means that the rate in the population is simply the mutation rate of that variant in the population. See how that works? So here S is saying how rapidly are we going to lose those alleles compared to the average allele. If we push it all the way to the boundary that it completely knocks out any chance of having kids, then that makes it simply mu. Okay? Make sense? Right. We don't see very many of those kinds of things. They're hard to see because they exist at this baseline rate of mu in the population. Okay, what about for other populations? So this is using the kind of so-called equilibrium population. And in population genetics, there's been this model, this kind of idealized model, where you have a, homogeneous population that is, abiding by all of the population genetics assumptions of things like random mating and that it's, endogamous, there's no admixture from outside populations, all those kinds of baseline assumptions. Well, this is under equilibrium, and now we can ask the question about, well, humans don't abide by our population genetics rules. It's very frustrating, right? So mating is not random. There's assortative mating. There are there's introgression from different populations, and it's all a jumble. It's all a mess. what happens to the combined allele frequency for, kind of typical gene in a sense, for other populations? And the answer is that it's essentially unchanged. Okay, and this is kind of weird, but let me show you a picture. So here's a picture. So this is selection coefficient, right? And then what we have here is the combined allele frequency. So the average allele frequency, the combined allele frequency for all variants in a given class. And what we have are a bunch of different population genetics models. So the, kind of bottom is this equilibrium population, this idealized population genetics sort of model of, 10,000 individuals, maybe reasonably consistent with what our population was like before the out of Africa migration and expansion. And then we can layer on a bunch of unusual population demography. So we can have, Icelandic population demography, so a kind of slightly larger population bottleneck, but smaller, overall, but I think more recent than the Finnish population bottleneck, which we think of as smaller but older, or the European population demography, so the, kind of go out of Africa and then have a massive expansion sort of population. Or we can do a variety of different expansion models. And all of these different models are trying to, in a sense, capture something about what we see with respect to, actual empirical data for patterns of genetic variation from these different populations. And this is the combined allele frequency as a function of the selection coefficient. Okay, so down here, selection is very weak. And so things are behaving more or less as if they were neutral variation. That is to say that genetic drift is the predominant population genetics force that is governing the frequency of these mutations, the combined allele frequency of these mutations. This is maybe a lot of where our kind of GWAS variation is, insofar as the kind of impact on phenotype is so much smaller than selection's ability to detect it, that we're really talking about things that look just like a normal kind of common variant. And then as you march up the selection coefficient and you get, to be more and more deleterious from a population genetics point of view, you see the combined allele frequency goes down. And it goes down almost identically for all of the different populations, okay? So this is one of those quirks of population genetics that pretty much regardless of demography, the overall burden of mutations in a given functional class, a given kind of selective class, is about the same, okay? This is, remember, combined allele frequency, so it's the overall burden. Now, let's distinguish that from what is happening with the individual allele frequencies, right? So this combined allele frequency is taking every single mutation that could be a putative LOF, summing them together and giving you a kind of group-wise gene-level sort of behavior. The combined allele frequency, the individual allele frequencies, they do look different as a function of population genetics demography. So here's our equilibrium model. This is why people like the equilibrium model. Everything looks pretty, right? This is a pretty looking picture. It doesn't look weird. You go to European population demography, it starts to look a little weird, right? Things are moving around. And that's kind of intuitive insofar as if you think about what is happening with a population bottleneck. Okay, so let's say we've got our effective population of 10,000 individuals. Now, let's say we have 100 mutations, 100 variants, each of which are 1 in 100 frequency. Okay, so we got 1% variance in our population of 10,000. Now we're going to grab 10 people. Okay, we're going to grab 10 people and we're going to shove them into their own little new population, and they're going to go and found somewhere called Finland 2. So they've gone and founded Finland 2, our 20 individuals. What has happened to the frequency of every single one of those 1% mutations is that they've been boosted. But we don't necessarily get all of those mutations coming through the bottleneck. So we've lost some of the 1% mutations, but for the ones that actually managed to survive and get through the bottleneck, they've now been jacked up in frequency. And it will have exactly the same bulk burden of that class of mutation, but it's now changed the distribution of the allele frequency. So the individual variants now have very different shapes. Okay, so here, if we think about Europe, what we're seeing, so let's go through this picture. So we got each of the various selection coefficients. Okay, so these are our S's, this, and then what we have, and on the x-axis here is the derived allele frequency. Okay, so that's the relative, that's the actual frequency of the variant in the population. And then this is the proportion of alleles below that frequency. So you can read this as saying, if I go to selection, if I go to 10 to the minus 3, so that kind of frequency, and I look at a selection of 10 to the minus 1.5, so this is a pretty strong S, the vast majority of my mutations are going to be below this frequency of 1 in 1,000 and under an equilibrium model. If I go over to Europe, it actually is changed it quite specific, quite aggressively with respect to that individual kind of demography because of the rapid expansion. Okay, question? Yeah, all right. Okay. So, the relevant range of selective pressure, and this is why we look at the individual level frequency and the combined allele frequency in general, is that it gives us some idea about what kinds of population genetic selective forces we're actually going to be looking at when we're looking at rare mutations for complex traits. So the relevant range is really this S between 10 to the minus 3 and S of 10 to the minus 1. So, and an S of 10 to the minus 1 is a very, very big S. Right? Make no mistake, that's a very, very large S in the scheme of things. And the, things that have weaker selective pressure than 10 to the minus 3, that's really the fodder for GWAS, okay, or CVAS if you prefer, right? So the more common variation will be soaking up the variability in the population in that kind of fashion. All right, so how do we actually think about S? And tried to give you some ways of thinking about it, but we can think about kind of taking a direct selection point of view. And this is what I might describe as somewhat contentious in the population genetics literature. Actually, we just don't have a very good answer to it, which is that nature of the relationship between what the impact of a variant is on a given phenotype and its impact on fecundity more generally in the population. Okay, so how does that actually play out? But if we think about the selection ranges of 10 to the minus 3 to 10 to the minus 1, if we have a purely direct selection kind of model, right? So we say something like schizophrenia has a 1% prevalence in the population, thereabouts. If you go and look at kind of epidemiology in Finland or in Sweden or in Iceland, in places where they have large amounts of registered data and have really looked at the kind of impact of these kinds of diagnoses on the relative fertility and fecundity, how many children you actually have across time periods, right? So you can take everyone in Finland, for example, between 1950 and 1959, ask, do you have a diagnosis? If you're born in that time zone, in that slice of time, how many kids do you have, and do you have a diagnosis of schizophrenia, yes or no? And you can get an estimate of maybe of a fitness cost of around 50%, give or take. It's kind of difficult to measure perfectly, but that's maybe what we get. So if you've got a really big effect size, so a 10-fold risk, that's going to have a really monstrous S, right? So this S of minus, or this 1.7 S that's a really big S. Type 2 diabetes, on the other hand, much more common, much weaker kind of fitness cost is the expectation. Again, a bit speculative. You can look at a 3-fold risk, and that's going to have a much weaker S in the scheme of things and will tend to be more common. For what it's worth, from an empirical look at genetic architecture, most complex traits are, at least with respect to the common variants, behaving very, very similarly. There's not a lot of distinction in terms of the genetic architecture. For some of the rare variants, there maybe is a little bit of daylight, but the vast majority of that daylight is really concentrated in very severe phenotypes, things like intellectual disability and other major neurodevelopmental things that are very, very bad for you. All right, so let's now, having set up this idea of looking at the frequency distributions, let's now get to the part that we're excited about, right? So we want to actually test for variation for a given gene. Like this is what we're all waiting for and what we're all excited by. So first and foremost, we can try and keep it very simple, okay? Just focus on the loss of function mutations. Really focus on these mutations where we expect the phenotypic impact to be identical regardless of the gene. Regardless of which LOF you have, the impact on the gene we expect to be basically the same. Of course, there are things that are being shoved under the rug, biology is complex, yada, 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 but again, it's a model, so your mileage may vary. Disruptive alleles are relatively pure, relatively homogeneous, relatively easy to interpret because the effect size equals the enrichment, right? So it's almost like we're doing a common variant association test where we can calculate the effect size directly. That's nice. And now the real key is the frequency of these alleles and the effect size. Those are the parameters that drive how big your sample size has to be able to identify a given gene. What is the frequency of a class of mutation and how big of an effect size does it have on the risk? And so this is the key figure, frankly. And if there's one figure I would encourage you to like try and internalize from the RVAS paper, it really is this figure. Because what this figure is showing is the frequency of disruptive mutations here. So this is our frequency of mutations. And then we've matched that to different selection coefficients, right? Because we're looking at this combined allele frequency sort of way. We're doing a burden test. We're lumping it together. You know, that can kind of help guide our hands. And then we have the number of cases. Now this figure is created by assuming that we have an infinitely large control sample. We don't really have an infinitely large control sample. You'll all be very sad to hear. I myself am very sad we do not have an infinitely large control sample. If we do this as equal numbers of cases and controls, all you have to do is basically double the number of cases. Okay, so that's a comfortable rule of thumb when you're thinking about it. But again, model, this makes like certain calculations and simplifies the math. Okay, so here we have different lines that represent the increase in risk, right? So this is now a representation of what is the boost to risk conditional on carrying one of these mutations. And this, you know, x-axis here is log scaled, this y-axis scale is log scaled, so it's, you know, like a log-log plot. It's a little difficult, but not too bad. But it's, you know, nice and linear in log-log space. So the way to read this is, let's say we've got a selective pressure of 10 to the minus 2, and you know, it's again pretty big selective pressure. That gives us an expected frequency of 5 times 10 to the minus 5 for a given gene. If that has a, you know, odds ratio of 20, then we need something a little over, somewhere between 1,000 and 2,000 cases if we had an infinite sample size of controls, and then double that if we had to do a kind of case control match design. Okay, that makes sense? And then this is now representing the different levels of effect sizes that we're looking at across the space. Yes?
This is cumulative in frequency you're using for
yes.
non-individual.
Correct. Correct. So, bottom line. If the effect size is small and the selection pressure is big, we're going to need very, very big sample sizes. Okay, so here's a nice kind of table representation looking at different lambdas. So we got our different lambdas over there. And then here's the different kinds of selection pressures that we're looking at. And you can get to like 2.8 million. So this like line of 2 here, you know, you got a big S and you're over at 1 million, and that's, you know, pretty nasty. We don't really expect there to be something, you know, if there's something a very weak effect with very strong selective purifying selection, then we really are sunk. So the things that might be more realistic, maybe, and I'll say this with, you know, plenty of caveats, are things with intermediate selection, maybe a relative risk of 3. Like this zone is where I think we might actually be in reality. This kind of area. And that's driven empirically from looking at enough of these studies rather than being driven by something that the model would necessarily predict. Okay, so that's like our LOF. You know, this is sobering. It is worth remembering that this is going to take, you know, a long time. And in truth, it will take larger sample sizes than it took for GWAS to yield. That is empirically demonstrated for schizophrenia at this point in time. I think similarly for diabetes. I think that, you know, kind of first wave of traits that have come off the rank that are, you know, kind of good, solid, complex traits, like schizophrenia and diabetes, we got a long way to go. Okay. We can maybe do a little bit better. So let's add in some missense mutations. Now, the tricky bit with missense mutations is that we have these neutral mutations. And if the majority of our missense mutations are neutral, then they will kill our power, right? Because we're going to water down our effect size. Every single neutral mutation you include in your group-wise burden test, that's just going to act as a bit of noise. And that noise will tend to make the cases and controls less and less distinguishable. It will tend to push together the, you know, kind of association test, and that really weakens our power, and we don't really want to do that. So what would be really great is if we could say with great confidence what missense mutations really matter. And that's why there are like half a dozen, maybe a dozen different let us predict which missense mutations are functional pieces of software out there, because people see real value. There is real value in being able to do this particularly well, because it will dramatically cut sample size requirements for, say, the analysis of complex traits. So a natural thing to do is, you know, we've just heard the first 20 minutes or so on the relationship between selective pressure and frequency. So a very natural place to begin is this kind of idea of an allele frequency threshold. So how well can you do? you can depend this on S, but the frequency threshold is still imperfect because no matter where you draw a frequency line, there will be a mix of neutral and null alleles, right? So no matter what you do, there is this baseline of rare neutral mutations in the population. And that baseline will exist no matter what you do if you only look at frequency. But you can still do, a bit better. And so this is a way that we kind of looked at that. So you impose some allele frequency threshold based on the, missense mutations. And then you ask, how much boost do I get to my kind of association kind of test information if I add in the missense mutations compared to just looking at the LOS? And, you impose different, frequency thresholds. And as you impose different frequency thresholds, the peak for where you're optimizing the inclusion of missense mutations is changing as a function of the selective pressure. So you have to push this frequency threshold rarer and rarer, the stronger and stronger the selective pressure is against a given class of mutation. This is all well and good if we knew what the S was, but we don't know what the S is. So that's, sort of makes things harder for us. So, there's some uncertainty. Any questions? Okay. The kind of strategies to deal with this, I would point you to Price Sinyev looking at the variable threshold approach. And what that does is say, well, you know what? I don't know. I have no idea. So I'm just going to test everything and let the data sort it out. But when you do that kind of, I'm going to test everything and let the data sort it out sort of kind of analysis, when you engage in that kind of association, you pay a penalty, right? So because you have to correct for the fact that you've tested all possible thresholds. And so there are, ways of doing that, but that's a convenient approach. Okay, so bottom line, if you knew magically what the threshold to look at, you can get, about a twofold decrease in the sample size. And that's if you have, perfect knowledge about where you should draw that allele frequency threshold. And it will change as a function of each gene, and it, again, is driven by this selective coefficient. So what about function, right? So there's polyphen, there's SIFT, there are all of these different kinds of predictions and scores that are used to try and help us against this idea of sorting the wheat from the chaff. And you again, at an overall level, if you assume about a quarter of the mutations that are missense fit into this class of being null mutations, then you should see, on average, about twice as many per gene of that class of mutation than you see at the LOS. Okay, so there are more bad missense mutations out there to find, potentially, if our comparative genomics work is correct, which I think is probably pretty close to correct for at least many genes. Then as a result, if we could just nominate just those ones, we could really have a major impact on pushing down our sample size. We could really, really cut our sample size requirements for making these kinds of discoveries of what genes are driving risk for disease in the population. So you could use maybe some combination of this idea of thresholding and frequency and thresholding using something like polyphen, and here's what you get. So here's the kind of relative value. This is our effect size of the class of mutations. And then depending on the demography, this is, one slice of the universe for the increase in sample size that we get. And then the dotted lines are if we do a bit better with respect to prediction. So if you add in not only this frequency threshold T, but you say filter which missense mutations are good and which ones are bad, and you have some ability to predict that, you can further decrease your sample size requirements by some non-trivial factor, something like maybe, 2.5. And this is, I think, what a lot of the community is doing. If you go and read the kinds of exome sequencing papers that are coming out right now, a lot of them spend a lot of time thinking about where do I draw my threshold for including variation in my burden test? What should I weight each of the individual variants with respect to the contribution to a kind of burden test or some of the other kinds of tests that are out there in the literature? Those kinds of approaches are the ones that I think we have to use because of the relative challenges associated with power and gene identification. All right, let's talk about population isolates, because I think there's been a lot of discussion about the relative value of population isolates, and certainly locally there's a strong commitment to leveraging population isolates and their unique population demographies for boosting our abilities to detect certain mutations. Now again, this combined allele frequency That's what we're interested in looking at a given gene. And recall that the expectation, so that is to say the average frequency of a given class of mutation, given a selection coefficient, is the same regardless of population demography. But it is possible, that we have this distribution, right? So rather than just having one gene with a selective coefficient of 10 to the minus 2, we might have 100 genes with selection coefficients of 10 to the minus 2. Okay, so we have this many such genes. And if we have 100 genes, we now have this idea of a distribution. And the idea of the distribution means that the expectation of the allele frequency is still the same. That combined allele frequency is still identical across all of our different population demographies. But what is going to change is the variability in that distribution. Okay? So we're going to have some of these genes where the allele frequency of those bad mutations at that gene-wise combined allele frequency is jacked up because it got, lucky in a sense. It's a winner of a bad deleterious allele that's going through the population bottleneck. Okay? So how does that look in kind of picture space? So this is so the way to read this is on the kind of x-axis, we have the frequency of null over the expected frequency of null. Okay? So that means that we're taking a look at that expectation of the combined allele frequency for a given gene, and then we're imposing different selection coefficients. Okay, so we've got a range of selection coefficients, and we're saying what is the frequency of null for a given gene over the expectation. Okay, and what you can see when you look at something like Finland is that you see these red lines dragged to the right. Okay, and this is inverse cumulative. Okay, so that is to say, if I'm here, basically all mutations, almost all mutations, less than, well, right here is 2%. So right here, 2% of my combined allele frequency genes, if there's no selection, should have a frequency of a given class of mutation that is, 4 times as common as the expectation. Okay, so on the x-axis, we have the individual gene over the expectation. So you want bigger means that it's much more common. So if it's like a 1% combined allele frequency on average, if we've looked at something that is, 5 times as big, then that means it's now 5%. And the reason that we're excited about that is that it means detection of an individual gene is much more straightforward. Okay, so we can look at Europe, we can look at Finland, and when you look at Finland, you have this big stretch of red. So things that have selection coefficients in the kind of 10 to the minus 3 to maybe 10 to the minus 2.5, that kind of slice of the universe, there are genes where the frequency of these mutations is much higher than we would expect based on the population demographics, or based on the kind of population genetics forces of selection overall. That increase in frequency of mutations of a given gene translates into much easier detection. Okay, so if there is a gene that has a 5% combined allele frequency, that is going to be a lot easier to find with the same effect size than a gene that has a 1% combined allele frequency than compared to a gene that has a 0.1% combined allele frequency. Yes. Oh, questions. Go ahead.
Expected frequencies for this local population or for everything?
For the local population. So this is showing what the distribution of individual gene frequencies would be, conditional on each of those genes having a selection coefficient of 10 to the minus 3 for LOS. Duncan, you had a question.
Why do the lines cross?
Why do the lines cross? It's confusing that, isn't it? I think it has to do with the way the demography gets modeled vis-a-vis. So these guys, right? What's happening is these mutations, there are the average, it has to do with the size of the bottleneck, right? So this is all relative to the expectation. The expectation of the weaker selection coefficient mutations is much higher than the expectation of the stronger selection coefficients. And conditional on being a stronger selection coefficient, you then get a bigger relative boost to the frequency. And that's why you can see these kinds of crossing lines in this kind of population demography. Does that make sense? All right. Whew. Make me work it out on the fly. That's kind of brutal. Thanks, postdoc. See if you get any nice projects. No, okay, all right. So those are the kinds of forces that shape what we would like to think about doing from a kind of study design point of view. And so the reason that these finished studies are really, really effective at gene identification is because they opportunistically boost the frequency. And we see this not only for, thinking about complex traits and thinking about the kind of selective pressures, but there's the kind of Finnish heritage diseases. Okay, so these things that are dramatically higher in frequency in Finland than they are in the rest of the rest of the world, and that is a consequence of that population bottleneck. So the same thing that drives up the relative frequency of some diseases extremely high, you can go and look at the Finnish specific kinds of diseases. It's not all bad though, right? And the reason that it's not all bad is if you look at the rate of cystic fibrosis, for example, that's much lower in Finland than it is in Europe writ large, and that's because the population bottleneck got, unlucky with respect to cystic fibrosis alleles, which is lucky for Finland, but they got unlucky for other diseases because, again, the expectation of the combined allele frequency of a given class of mutation for a given S is the same, pretty much independent of demography. All right. So that's some of the study design considerations, things that we think about when we're trying to do these tests. Those are the sorts of questions that are still, I think, fair game and still very much under development. We are not perfect at predicting the phenotypic consequences of a given variant in a given gene. We're actually pretty lousy at that, I think, still. We're getting better, but there's still a lot of room to go. You will hear a lot more about other ways of leveraging kinds of patterns of genetic variation in the population if you come to Caitlin's primer, where she'll talk about constraint and I think maybe de novo analysis, or Daniel's when he'll talk about nomad and exag. Okay, one other thing from a purely statistical point of view is this idea of dimensionality. So a lot of people are running association tests and getting QQ plots that look like this. So here we have our typical QQ plot. We've got our minus log 10 p-value on the x-axis, and then we have the minus log 10 p-value on the y-axis. This is under expectation. This is what we observe when we do our analysis of the data. And you get this sort of, I'm sad, right? This sort of, I'm not going to quite make it, I'm an impotent QQ plot. And it's actually a little bit different for different tests, right? So we have our, logistic regression wall test. It's really not performing very well from an asymptotic properties point of view compared to the allelic chi-square and the kind of Armitage trend test. Now this is looking at 40 minor alleles in 2,000 individuals, and then we can think about looking at maybe 80 minor alleles in 10,000 individuals, and things get a little bit better. And the intuition here is that the rarer a variant is, the more limited the range of possible p-values you'll ever even observe is. Okay, right? So if I've got a 2-copy variant, it is very, very difficult for me to get an extremely strong p-value out of that kind of variant if I do a straightforward case control association test. That is not actually going to do the job from a case control variation, case control association test. And so that's why this idea of rarity turns around more the number of the copy of alleles rather than the sample size per se. So by the time you get to maybe 80 copies of the minor allele, you're reasonably well distributed if you use a reasonably sensible test statistic out to kind of 10 to the minus 8. And that gives you some kind of confidence. And so that's not a bad rule of thumb if you need such things. Okay, now just to close, I'm going to talk a little bit about data and a little bit about some of the things that we're developing in the lab that might help with the actual analysis and QC of these activities. So again, we're talking about sequencing. We're excited about this comprehensive capture of genetic variation. Data generation is accelerating, so this is data generated at the Broad through, the beginning of this year. And, this is a pretty, steep curves, right? I mean, these things are pretty gnarly. We're doubling our data size every 8 months, every 9 months, something like that. And that's kind of a lot to kind of keep track of. Here's what our QC pipeline looks like in the lab currently. So these are all of the things that we look at when we perform an association kind of analysis from a sample and variant QC point of view. Hat tip to Andrea Gana, another postdoc in the group, who has put together this sort of plot of all of the various things that we look at from sequence quality from, contamination and chimera and insert size distribution, all the way to sample and variant QC metrics, things like Hardy-Weinberg or call rate or allele balance or any of the other various things. And this used to be very slow. So like a year ago, this was, day of compute, 2 weeks of actual real time, very painful, not very satisfying for anyone, very, very difficult experience. And it was a lot of kind of chewing gum and, pipe cleaners tied together. It was almost like a kid's arts craft project because you had to string together all these different pieces of software to actually arrive at any sort of coherent kind of QC positive analysis-ready data set. And so a little over a year ago, I brought these 3 guys on board and then teamed them up with these 2, and then they brought on Dan as well, and they now kind of compose this team Hail with Cottonseed as the technical lead. And the whole model that we've adopted in terms of actually executing these kinds of QC and analysis things is to change the way we think about actually analyzing data to keep pace with this challenge of scale. So when you do any product development of any kind, and we can think of this as product development at some level, the best thing to do is go and interview your customer base, right? You do your little testing to figure out what people want and what they don't want. And being here at the Broad, it's really great because there are, dozens upon dozens of computational biologists and bioinformaticians that are interested in actually driving forward our understanding of genetic disease. And so they did a bunch of interviews. And then the other thing that Cotton really pushed and was the real main architect behind that really is changing the way that we analyze these kinds of data sets is that we moved away from the old model where you have your high-performance research compute cluster, a big data pile, and then you shovel your data to your computer. And instead, up front, you break your data set into lots of small pieces and then distribute your data set and perform the calculations in parallel. And that kind of approach, this distributed computing approach, buys you the most important thing, and that is scalability. Because right now, we're doubling our data every 8 or 9 months, and if that is the continued arc for a sustained period of time, the old model breaks. Actually, it was already broken in truth. And so this potential to just add compute resources as we need them and scale, maybe not indefinitely, but at least sufficiently to keep pace with our current data needs, is really, really important. And that's all through the magic of the Apache Spark big data stack. So using Scala and Spark and then a whole bunch of other tools that you can read about up there, they went and started to build this toolkit called Hail. And they're mathematicians by training, so they also do some work on kinds of algorithmic development as well to further, streamline our processes. Now, in the first 6 months, we moved from 2 weeks to 42 minutes to maybe even 9 minutes if you use a larger compute cluster. And this is a sustainable kind of improvement in speed, at least at this point in time. There's still, another order of magnitude or 2 or even possibly 3 of speed improvements that Cotton thinks he can kind of squeeze out of the toolkit, but nevertheless, it's now possible to take these kinds of data sets and actually almost get to real-time sorts of QC and analysis. And I think that's really important, and that's what really drove, Nomad. So you can go and look at Nomad that Daniel MacArthur and a boatload of other people have put together, and the kind of QC and curation of that data was all facilitated using this this Hail approach, and that's now being served up for the broader community to use, this combining of genomes and exomes as we get there. You can learn more about it at hail.is. There's a lot of outreach. There's the group chat on Gitter, which is a kind of GitHub pull through, and you can sign into this right now and take a look if you like. So with that, I'll conclude because we're just about at 9:30. But just to flag a few other things in rare variant analysis that if you're excited and enthusiastic about this space and want to learn more about some of the various projects going on institutionally and some of the additional approaches we use scientifically to, again, drive forward our understanding of what genes drive disease, I recommend both Daniel and Caitlin's talks. I think both of them will do a really excellent job further building out some of the approaches that we have for these kinds of considerations. And with that, I'll say thank you and say we have maybe a minute for questions.